and then they create this, it's called quorum sensing. So they multiply themselves, and they can also interact and then also cooperate. That's really happening in the inside the, in the nature, in other words. So when we look at the
all I want is nothing.
Man, me and Jesus are cool.
functions, and they can be regarded as molecular transmitters. And so that's the typical eukaryotic cell case. And this is a well-designed eukaryotic cells. The prokaryotic cells, like the E. coli bacteria, is more irregular behavior, structure, as you see here. But the same thing, like little computer, right? So it has this nucleus and plasmids. Okay, it's like, can be seen as a memory. Like you can see here, memory. You can store communication functions, sensing functions, and actuation functions. So you can program them, as I told you before. So we can have coding, and then we can code them into this memory, and then they can do what they want. And so then also these plasmids, they can be regarded as processors. These are the pilies, and they act like sensing and actuating. And this flagellum, like a tail, is like an actuator. It navigates the bacteria. And, they're extremely slow moving. Like, 1 centimeter, they need like 4.5 hours. For 1 centimeter plus, they're always tumbling, so slow. And what you do is you feed them with sugar, they grow exponentially. And then, it's moist area, of course. They move around. And then if you don't feed them, they don't have enough sugar or glucose, then they die, So that's the typical bacteria behavior. Then there are these receptors and membrane, cell membrane. They're like nano-power unit, right? Energy sources. So now this is a typical mitochondria under the microscope. This is a real picture here. So I told you the energy source for the cells, for prokaryotic or eukaryotic cells, especially eukaryotic cells, is the mitochondria. So what happens is that they combine the glucose, amino acid, fatty acids, and oxygens, and synthesize as ATPs, or adenosine triphosphate. So now by this way, the energy is loaded, harvested, you can say that. For example, so when you are tired, you say, let me go and get fresh air. So that's typical, you need oxygen, right? So that you can, charge your mitochondria. Or when you have little kids, the doctors tell you to not give chocolate or sugar before they go to bed, because all the mitochondria will be charged, they will be hyperactive, right? So that's the typical example for the energy sources. Then you can see also for the biological memory and processor, like you can see these DNA strains, right? And DNA strain in the nucleus contains the information of protein structure, right? Like it can be seen like this. Some of you may know this Turing machine, right? One band Turing machine. So like that's the memory, right? And the ribosomes read and process the DNA information from here and synthesize the proteins, okay? And the proteins, looks like this, control the functionalities of a cell. Like, for example, for transmitting, it's called cell signaling, or receiving, it's called ligand binding or chemical binding, right? So these are the terminologies that the biologists and chemistry people use. So as I always kept telling you that there's communication between these cells. Like here is an example, eukaryotic cells, they release these molecules, as you see here, like they can be proteins, ions, and hormones, and like they flow, and then, normally they're in confined area, right, inside the body, but you can also see that that's like, if you want to see as a model, you can also do free diffusion, right, free space. But anyhow, this is I'm trying to explain the basic ideas about how molecules are flowing, and the molecules can carry messages, right? And then the receiver will receive them. You will see the process. And then the prokaryotic cells are like this. So same thing like this. And it releases these molecules, for example, auto-inducer exchange for quorum sensing, Or there is also another way of doing communication is that so like this bacteria will upload the information from this device, any other nanoscale device, any type of device as transmitter, and it will be loaded up to this plasmid, and then it moves slowly, and then it can come to the to this one, and then or to this one, and then it can upload the information from here to this one, kind of like handing over the information to the next one, and then finally it reaches the destination, right? And this process that one bacteria gives to another bacteria the contents of its plasmid is called chemotaxis process. And then when they reach the destination, as you see here, they eliminate. Indeed, under the microscope, we can see that. So it's called also mating, that the bacteria are mating. They are very happy that there is a elimination, right, like the light. And so that process is called conjugation. So this is the typical, that's what's happening in the nature, by the way. So now you can see also in this picture how the transmitter looks like. Like here's this one eukaryotic cell and all these molecules, or if you have one type of molecule, you can call them particles, they diffuse, okay? And that's why you can use Brownian motion to model them, or diffusion solutions from Adolf Fick's physics laws 1 and 2 to understand and mathematically model these processes. That's what we did, and other people do that too. So now they're releasing, and they have some messages on them, right? And then they flow. And of course, some of them you lose, that they're typical losses. Some of them will reach the receiver. And then as you see here, when they reach the receiver, and they are sucked by the receiver here, as you see here, by the gap junction, and that's called particle binding or ligand receptor binding process. So, like their binding, are received by the receiving end. So this is the typical behavior of the receiver and in the molecular communication. And that's why these are called also chemical receptors. Right? And some of them, of course, they cannot reach, they move somewhere else, right? So, or just they're lost. So these are the typical transmitter receiver modeling of these eukaryotic and prokaryotic. Years, or was in the past, can we create man-made biological nanomachines, right? So I explained so far the nature. So can we create these man-made, programmable or genetically engineer these machines? The answer is yes. And we can do DNA manipulations and genetically engineer them. So that's the good news, right? So now you can see here the prokaryotic cells, the bacteria is well understood. So we can genetically engineer bacteria without any problem. We can also have codes, we can program them, and they can do whatever we want. So that's really like well understood, and people are applying this. For example, I have many applications here. One of them I remember, I don't have the slides. There is so-called Crohn's disease, an awful disease for the intestines. So there's no cure for it. And there's a bacteria-based disease. You have to go to the toilet all the time. You can never cure, unfortunately. But with this technology, people are trying to use genetically engineered bacteria, like healthy ones, and they inject inside the body so that the healthy ones will fight the sick ones or ill ones. And the probability is very high. For the timing, what they do is they take the feces of a healthy person, and then they have these healthy bacteria from a healthy person, and they convert them into pills, and then people are taking them. But that's really only 20-30% success rate. But with this, we are hoping that it will get very high cure rates. Now, this is just an example. Any bacteria-based type diseases, we can fix them, right? So now the human cell engineering is really not easy. Otherwise, it will be fantastic, right? So we can solve all the illnesses in the human body. So now, how they can try to do is they try to insert a piece of new DNA code into so-called lentivirus cell. This is a special type of virus using genetic engineering, and this lentivirus infects the eukaryotic cell, the healthy cell, and injects a new DNA code into the cell DNA right here. And then it replicates itself, and then it will bind itself here, and then it can go and attack these, whatever the problems are. So, and this is still ongoing issue by these bioengineering medical people, etc. Right. So now you can see the objective here is we try to create man-made machines, like these or these here, right? Like, man-made meaning, genetically engineered. And so the origins go back 100 years ago by Paul Ehrlich. He was an Austrian doctor, medical doctor. He created this concept of magic bullets, in German magische Kugeln. And so he received a Nobel Prize in 1908. These agents or these bullets will go and kill the targeted diseases, and without affecting the other healthy parts of the body. Right? Otherwise, you will die if it has all the negative side effects. Right. So, what is the lessons we will learn and get? So, this picture was done in 2012 in Finland. There is a 6 pages report about that center that I created at that time. And so the guy made a photo of mine, and then he put me at the wall, and then he put this in the newspaper. So he had all this in Finnish, of course, right? All these things. That we can have solutions for Alzheimer's, epilepsy, depressions, then brain cell monitorings, any tumors you can have, and diabetes, for example, heart diseases. We can always monitor them with special design and program these devices. And then, cancer monitoring devices. All of that, again, depending again what you are trying to, monitor, right? And so you can inject these magische Kugeln, or now they're called nanoscale bio-nano things, and then they can observe what's going on. So as you see, let's say you have a heart disease problem, they can always monitor it. And if there is any alarm, they can try to solve themselves. If not, then they can alarm to outside through using this gateway, and then go to the physician, and the physician can reprogram them, etc. Right? So this is really a breakthrough that that can extend a lot of years, right? But I assume we'll have some other problems, like here this COVID-19, right? It was expected, by the way. But anyhow, let's continue. So now we also did a lot of work with my former student, Youssef Shahibi. He was from Morocco. Now he works for a company. And Massimiliano is professor, as I told you. And this guy was from Samsung, a Korean guy. We tried to create this model for a particular drug delivery system.
Wow.
So you can see that, normally, I'm sure you learned this in the systems control theory or whatever. So you have these boxes, right? Input, processing inside the box, and then outside, right? Delivery. So in other words, you inject these drugs, medicine, and they travel through the body, right? And then, like here, you can create, and then they go to the target area, and then, they try to attack those ill areas to have cures for them, right? So the issue is the following. So now here's the target that, once these drugs reach this target, they lose their strength because they travel through the body, right? And then as they travel, they lose their strength. And then once they reach here, they're almost like half strength, having half strength to reach this target location, right? So now in this study, and also as you see here, this is kind of a communication network, right? Like a propagation, all these particles, drug particles are moving inside the body, right? It's kind of like a communication network. So we developed these as a communication network. And then it was a transmitter that we are trying to inject these drugs. And this is the receiver, meaning the target location will get these particles. And now that was the model, but then what we did is we tried to do it the other way around. That means we want to have 100% strength here, right? And then we go backwards and try to find, usually in the systems theory, what you do is control theory, input inside the box, you try to optimize the output, right? So we did exactly the opposite. So the output is the optimum value for this strength of the drug, And then we tried to find out backwards how much drug particles we should inject into the body so that we will get the highest degree of the strength for the drug here. So that was really a good paper that we published some years ago. And then we also received this, I call this Monaco Project, Molecular Nanoscale Communication. These were professors in Georgia Tech. He's in info theory, and this guy was from mechanical engineering. This guy was from biology. This was from computer science. So I created this team. We got 5 million in 5 years, 11 to 16. We had 20, 25 people working on the project. And although it's over now, so the project page is still there. We published many, many papers, and those papers got a lot of citations. So they were really interesting. And still people are using those, bacterial communication. What we did is, so the idea was we tried to establish theoretical foundations of molecular communication networks, understanding how they communicate, and try to map them into the communication networks, communication theory. And we developed this fundamental base for that and for the bacteria-based communication. And then we looked at network architectures like modulation schemes and communication protocols. And also we published them, modulation, medium access control, etc. And also like the collisions, right? All these bacteria collide, by the way, and losses and all that. And then, so the NSF wanted that we will implement everything NS3, which is a publicly available simulation tool. So we also did that part. So this was all theory, like academic. And then the other team, the mechanical engineering and also the biology guy, we did physical experiments with actual bacteria. Of course, here in the top theory, you can do anything, right? You can conquer the world. But when you have these physical experiments, then you have to reduce all the theory so that, you can have some experiments to validate some of these, theoretical solutions. And that was quite successful. As I told you, we have many, many publications. So now, and then I wanted.
Can't believe that.
And then we wanted to have an application. Right? And then we called this panacea. Panacea means in English, a remedy for everything. That's a Latin word. I found at 3 o'clock in the morning when I was sleeping, I got up, we call it panacea. And so then, we started in 2017 with all these meetings and projects, papers, and then finally we published that. So it really takes forever, when you have all these members. So now the issue is how can we detect infections early for critical patients, like patients in critical conditions are better. Like, for example, the cancer patient, like chemo patients, they're very vulnerable to serious bacterial infections or cystic fibrosis for the young children. Infections come wave by wave for these young children. And then, so our solution should somehow early detect them, mitigate them, right? Eliminate them. So in other words, we try to continuously monitor of these bacterial infections and take actions, right? Early detection, timely administration of antibiotics, and also tracking efficiency of the antibiotics. That means overall, we try to reduce the mortality and also the hospital costs, right? So, like I remember, it's a bacteria base, by the way, diphtheria, like kuş palazı in Turkish. And I had it, I was 4 years old. And those years, Turkey was not like now, right? So I remember there was another little fellow next to me, Musa was his name, he died. I remember the parents were crying, Musa, don't die, Musa, die, in Turkish. And so then doctors told my parents that, either Allah will help or you will die too. And then somehow out of blue, this thing came out, this black thing, from my throat. Kuş palazı is like, it's choking you, right? It's typical bacteria base, So really, it's a serious thing, as you know. And now you have shots and aşı, etc., but still. So now what we're trying to do is, so this is the Internet of Bio-Nanothings, right? So this part is the human body, like physical world. And you can see here, this word also remor, I came up with that in the 3 o'clock in the morning. Like istihare yatıp having some ideas, right? No, but yeah, remor means explorer, So these are BioNano things, like BioNano sensors, which we will inject inside the body so that they can monitor all these. Of course, they will program, and they can monitor these bacterial diseases, right? And then you see, remember, we have these gateways, and near field communication through cell phone, we go to the cyberspace, right? Cyberspace, this part, right? Cyber-physical space, right? So physical space is here, cyberspace. All this data will be collected, right? They can be aggregated when they have similar data. We'll process them. And then we have to, of course, consider the security aspects, the privacy issues. And then we will use, I will explain you more, some machine learning algorithms to handle all these collected data. And then you can see, like CDC is the Central Disease Control here in Atlanta for the entire USA. And, we can help them here, or clinic databases or CDC databases. So that's the closed loop of the Internet of BioNano Things concept, right? And so this is the entire project that we had. You can see we have the Trust One physical domain, like this part, like hardware design. And then, of course, we have all the interfaces. You will see them, and integration of these different components of these BioNano sensors. And then also look at the physical layer hardware security, right? And then the communication among these hardware devices that we developed, like how do we model, that's more communication theory, right? Infections and drug deliveries. And then how do we realize the near field wireless and power transfer between the BioNano things to the gateway, and then from the gateway to the cell phone, and cell phone to computer science type research. How do you collect, aggregate data? And then how do you use machine learning or deep learning or any type of learning algorithms to help us to early detect these infections and process them and then take actions, right? And then actions meaning, for example, deliver some antibiotics or drug delivery, or also visualization of the problems. Of course, all the security and privacy are important when we do all of that. And then, these are all the trusts or work packages, as Europeans say. And then here are the experimental validations, testing individual components, then prototyping Panacea in vitro and also in vivo. So that's why we had these, not only 5 professors, and then we had also like 10 different people working on this. So what are the challenges when we look at the cyberspace, right here, this part, right? That's typical all computer science related research. So how do we effectively collect data and deliver to the computers or cloud or to the users? And we may have energy and transmission constraints. We have to consider those. And bacteria concentration reflecting infections may vary from patient to patient. So that means how can we early detect these possible infections with high accuracy? And then also, how can we quickly detect those infections and with high accuracy, right? That also means how can we relay or transmit these data securely and privately to the physicians, for example, or the clinics, right? Oops. And so then, here is more. How do you collect, aggregate sensing data here? Deep learning for early detection. Learning-driven drug delivery. We can have active drug delivery, meaning automated antibiotic and enzyme delivery by implanted or dressing devices, or passive drug delivery, meaning alerted patients or patient takes antibiotics orally by the suggestion of the BioNano Things platform. Then how do we visualize a reliable platform to access and analyze data?
Monitoring us
As you see here, it's all
24/7.
data processing on this side.
In our
And then
smart
we also
brains.
must look at the hardware embedded security,
Smart cities,
issues,
people look
authentication,
up the real
confidentiality, integrity, etc.
time
And
by
also develop privacy-preserving data
60.
storage and processing capabilities, right? So now you can see here that physical domain, I'm going down now about these ReMore. So ReMore has, we also call this BioNano thing, right? So it has these 2 parts. That's called BioNano sensor. Like they have, like they can sense quorum molecules, and there are these reporter bacteria, membranes here, and antigens and antibody here. I will explain to you more about the details of this. So that's the infected area, for example. Because infected area, these bacteria are releasing these molecules, right? And these sensor must somehow capture those process them and send this to the interface here. And that's, again, I will also explain that to you. So these together makes one bionanothing, I agree more. And this is the skin. We're using magnetic induction communication to the gateway that we had this, like either the like tattoo type device, like here, Band-Aid, or a watch, right? And it goes to the near field communication to the cell phone, and then cell phone can use the internet backbone, transmit to these doctors, etc. clinics. Right? Now let's look at the like more detail here in this part, right? So that's more technical. So what we have here is, in this case, we have three different sensing types that we are, or sensing units of the bionanothings that we are planning to design and implement. So one of them is called electromechanical sensor, as you see here. So there are functionalized electrodes directly bind with quorum sensing molecules, like here, right? So that's the first one. The second one, it's a bacterial sensor, right, or sensing unit, an engineered reporter, like here, some of bacteria like this, okay, and produces an electroactive product upon reception of these quorum sensing molecules, okay, and this electromechanical sensor generates currents corresponding to the concentration of the quorum sensing received, and then they're again, you can see binding process, ligand binding, so they're binding or received by this transducer here. And then the third one is called bacterial sensor, okay? So an engineer reporter, bacteria produces bioluminescence, right, like these photodiodes, or fluorescence upon reception of this quorum sensing molecules are coming here, and they're exciting. This is a porous membrane. They have these holes, and these bacteria can pass through, and the bacterial sensor will capture them, right? And then we apply these, as I told you, bioluminescence or fluorescence, right? And then a light sensor, like here, generates currents, right, which correspond to the concentration of the quorum sensing received, okay? So now what happens is we maintain live bacteria inside the sensor here is difficult, and the sensing process is noisy. So that's one of our challenges that we are trying to solve this problem. So, that's the, keeping the bacteria alive in this, inside the sensor is the biggest challenge here. So now the second case is the designing of the sensor interface electronics, right? Readout chip, like here, sorry. As you see, I mentioned this here, readout chips here. So now we can have these electronics to process signals. I told you that we are using the magnetic induction, like these are the coil antennas. There's the skin, and then on the other side, on top of the skin, that the Band-Aid type or tattoo type thing has all these components inside, right? Like typical, like passive device that can receive this from the readout chip. And then we can also have these wireless simultaneous power and data transfers. And then what we have here is, in this first one, is 3D rendering of a solenoid coil and showing its key geometrical parameters, meaning how many turns do we have, what is the diameter of these turns, like all of this needs to be optimized, right, determined. And then we have this B lump RLC, the resistor, inductor and capacitors, right, model of a solenoid coil, and showing its parasitic resistance and capacitance. And the third one is the example bonding wire coil for the wearable hub. So we have also this, these are all physical part, right? And then I told you this before. So this part I explain, and then they go out, and then they go through the backbone to the doctors and physicians and clinics, right? So now the IOBN components, we can again model them for the communication aspects. We are in the middle now for these Trust 2. So we have this like typical transmission process, reception process, and the propagation, right? The channel or the communication network, right? So these bacterial infection, these molecules are coming out, and and then they move right inside the body, and then inside, right, and then the remor, which is closed. I'm showing you this one, right? So now, like from here, transmitter moves here, and then it will be received by these remors, right? And then the reception process. So then we developed these molecular communication channel model and analyzed it. And then we realized that there's a typical communication theory, right? The channels are time varying, and there is this diffusion process, right? All these bacteria is flowing around within cells or between the cells, and they are shadowing by the tissue structures, shadowing problem. And then there are also multi-path effects, like typical wireless channel effects, right? And you can see here, we can also use them as a MIMO type communication, like MIMO multiple input, multiple output. Like this is one side infected, another side is kind of like two areas, like two antennas. And then these are the two receiving ends, like remors, like 2 by 2, right? So, and then we can, we analyze this also, understanding this. So experimental validation, we had testing of these remor components, each individual bionanosensors and also the chips.
Data.
We tested the variable hubs. We also tested communication protocols. And then we tested all these aggregation algorithms on the cyber area, the learning engines, like deep learning engines, and closed loop with passive delivery, like transmitting these drugs into the body, right? Passive drug delivery, and also security protocols, etc. Right? So the simple, experiments we did here, as you see here, that we had control release of quorum sensing molecules into these body type fluid here. We place this remor inside this tube here, so that this remor will measure these molecules, which were infected or not infected. That's what we are trying to find out. And that remor, the bionanosensor, send the value, or is the bacteria is infected or not, for example.
Quantum
and then we also
technology.
try to mimic the human body. You see a steak here. That doesn't mean that's a real
What
stomach.
about the
So we try to
privacy?
mimic the human flesh, right? Flesh. So that, we had this, like skin, right? The human body. So now same
Good
concept.
God.
So now it goes through the human body, right? Through this meat, and then through a variable hub, and then reaches this here. Again, try to measure the quorum sensing molecule concentrations and find out
Disgusting.
from the concentration level about the bacteria status, right? And then we also did experimental validation in in vivo testbed. My son Guanlu, professor, he had these with these rats. So we had knowing sublethal dose, doesn't kill, but it's infected. We put also the remor here, and then lethal dose. We can, we try to get the information from these remors through this to the back. So I said, why don't I have another smaller version of this project there? We call it Clipeos. And we submitted this proposal back in November. And then in March, we received that we will receive $1.8 million for 4 years. And so now, of course, everybody talks about now diseases, right, etc. Now, a lot of people are starting to work on these issues. So I had these since many years, right, these ideas. So now all these will have different design. We actually we need people. So if some of you are interested, so how can we detect that? So they have this Pseudomonas erythritidis to do for this pneumonia detection. And so this was lactons and in vitro homoserine lactons, like two cooperation of quorum sensing molecules. And these molecules are found in bodily fluids such as sputum and blood, and detect in such a way we can detect these modules, right? That's the question. Like the bacterial density, transmission process, propagation inside the body, and then diffusion process in porous medium. So now you can see here, this figure shows the diffusion process in porous medium. So the black dots are representing the cells, right? Parts, that's pores, like, if we will not have these black dots, it will be straightforward, right? It will be much faster, as you can see here. But unfortunately, it's not the case inside the body. So they will be moving like this, right? And it's also, this area is wet inside the body, right? Wet medium, it's called. So then the molecules are flowing through these pores. And then we are showing this direct flow versus the reality. and it takes much longer of the
channel.
human body, like
He's
from the
found
side.
that and he's
You know, this
showing it up.
infection is, the concentration is very high. That's why you have this red area. I'm
Wake
sorry,
up
the wall
the
infection,
screen,
I
come on.
the higher is the concentration we can
Wake
capture
up.
that. So that's the lessons we
Humanity
learned
at
from
risk.
these studies. Oops. And then, so now another application is about, for the military personnel, how to be capture these Acinetobacter baumannii infections. Again, they're bacteria based. So they're like kind of like bacteria. They create, find the cure.
Tap
and
the
this is the number one
screen.
pathogen
Come
required
on,
new
you
antibiotics
idiots, wake
by
up.
the World Health Organization.
This is your...
Come up with in situ lab on a wound system,
You
log
don't
system. That's what
care,
we try
that's
to
your
do.
problem.
And I will skip because it's like a band-aid tap, right? It looks like this.
Biosensors.
When the wound is here, then as I showed, reporter bacteria, B, and the light. Again, I explained this before.
You
so
all
now,
go
again,
to
all
sleep.
these things, I will go to the last
Well, I
part
ain't gonna
because the
show
last
him
part is
sleep. You're
interesting
gonna
to talk
be
about. Lab on a wound modules, integration, experimental testing, then cell to reproduce. Otherwise, they die, right? So that's why I kept telling you. And some bacteria are beneficial. We all absolutely not. So bacterial infections can be cured with antibiotics. Viral diseases, or they give you steroids, right? But that's not a cure, right? So most areas, Vibrio cholera, and all the other ones are bacteria-based. I mean, I gave also 3 of them before. And here we have HIV, influenza, and SARS, and COVID-19, and all that stuff, right? So these are typical examples. That's the problem. So the most reliable virus detection in laboratory is done by this PCR process, polymerase chain reaction.
Share it out.
So, so there's this body sensors. This is a huge area, that's antigens. And then an antibody tags a virus or an infected cell for attacking by other parts of the immune system or can neutralize its target directly. So these are the 2 sensors we have in our minds to design and come up with, or, they're, maybe off the shelf existing, but I don't know, but they're not used inside the
I'm not watching anymore.
So now, so we see that COVID-19 has all these challenges. So we need to somehow do these, luciferase sensors and
I can't watch that. This demonic times we're in. I just know what they're dealing with. People are so asleep. The souls, the demonic souls, they're not dealing with the flesh and blood. Let's get off that and get
have lost that
One last adjustment.
It's not going to be entertaining. I've got to turn my electrics off. This is demonic times. I'd always known there's something, they're testing on target individuals. This is demonic. 6G.
Oh, happy birthday to me, 25th ninth, 2023. Despite those powers that be, I am still here. To my family that I don't see, you know who you are, and to my grandchildren and my son and daughter that I don't see because I've been taboo for years. At least I'm not alone. My wish is humanity, no matter what happens, moving forward. I know that just because we're all different, on different journeys and paths, whether you're blood family or not, we will survive. I was born in this timeline for a reason. I'm still thriving just because I'm run down as well, and these frequencies around me.
Check my YouTube channel.
so can we create
Watch out. This demonic times we're in. I just know what they're dealing with. People are so asleep. The souls, the demonic souls, they're not dealing with the flesh and blood. It's obvious, isn't it? No matter what you're trying to get off that.
You have lost that.
One last adjustment.
It's not going to be entertaining. I've got to turn my electrics off. This is demonic times. I'd always known there's something, you know, they're testing on target individuals. Systematic 60.
Oh, happy birthday to me, 25th month, 2023. Despite those powers that be, I am still.
Discover more with Marriott Bonvoy.
Surveillance. It's not about bloody speed. My phone's on battery's low. I've got to charge it up. I'm going this life now, thanks to my fucking. Take care.